Hello everyone. My name is Joe. I'm a postdoc in the School of Biosciences working with Marco Catoni. And today I'm going to share with you a collaborative project called Sounding the Epigenome, where it's a collaboration between biosciences and music, where we attempt to engage some people, or I think of it as engaging with other people in the science that we're doing through a piece of music which was composed using epigenomic data from oak trees. So I'm going to split my talk into three parts. Firstly, I'm going to talk a little bit about the science that went into this composition. Secondly, I'm going to talk about the composition itself and how that was created. And finally, I'm going to play the piece, which was actually at a concert at the university in March earlier this year. And and see what everybody thinks of this piece. So the science first. Here we know, So we know that plants form memories of stress. So if you have a plant which is under some kind of stress, in this case possibly drought, after recovery from rehydration, this plant develops a memory of the drought event. And if it is then subsequently challenged by the same stress, because it has a memory of the drought event, it actually is better able to survive this event, and repeated stress results in repeated improvement of the plant's resilience to stress. Membra, which is the project that I'm working on, is particularly interested in how trees are able to retain, acquire, and transmit these stressful memories. And we think one mechanism by which this happens is through epigenetics. In this particular case, epigenetics is the modification of cytosines in the genome of a plant with the addition of molecules through particular mechanisms, which results in an inheritable and environmentally controlled change that then causes the plant to differentially express the genes that this region of DNA is encoding, and therefore result in physiological responses that will allow the plant to better survive stresses that it has a memory of. So, as has been shown earlier, we leverage the BIFoR face facility, which has higher levels of carbon dioxide in its arrays. And this simulates the elevated levels of carbon dioxide we expect to see in the next few decades because of human-driven climate change. What we did was we took 23 trees from the BIFoR oak trees, from the BIFoR face facilities, with 12 under elevated conditions and 11 under ambient carbon dioxide conditions. We collected leaves throughout the canopy in over 3 seasons in 2022. And we looked at their epigenome with the idea of trying to better understand how the epigenome changes in response to memories of elevated carbon dioxide. We were also interested in the offspring of these trees because as I mentioned before, we expect the epigenome of trees to be inheritable. And because these oak trees are long-lived species living up to 1,000 years or more, the relatively short spike in, hopefully, relatively short spike in human-made carbon dioxide, if it, say, peaks in 2050 or 2060, the carbon dioxide levels. Whatever effect it has will persist likely in oak trees in this country for decades or centuries to come, particularly if, of course, the trees pass these epigenetic memories down into their offspring. So what we did, we got those leaves, we lysed those cells, we removed proteins, extracted the DNA, and then we sent it off to a company to do some whole genome bisulfite sequencing for us. This is the process by which we identify the cytosine methylation status within the oak genome, and they return something like this. Now, the oak genome is over 800 million base pairs in length, and we have 23 of these trees. So this is a very small extract of hundreds and hundreds and hundreds of gigabytes worth of data. If you look closely at the bottom along here, you can see the sequence or part of the sequence of the oak genome. We take this data, we compare the elevated individuals with the ambient individuals, and across all these 800 million base pair locations, we look for regions where there is elevated cytosine methylation in one condition or the other, and we do some statistics and some processing, and we end up with a large list of differentially methylated regions. So regions within the genome which are differentially methylated between trees in the elevated and ambient conditions, and this represents essentially memories that have formed because the tree has been exposed to elevated carbon dioxide levels. Now, here I'm going to showcase a small example of some of the leads that our science is taking us, but this isn't a science talk, so I'm not going to say too much more than just to give you a flavor of some of the leads that we are pursuing in the science. So at the top, along the x-axis is essentially the position. So remember, this is the oak genome. It is 800 million base pairs long. This is a small fraction of it. And the blue bars you can see are the positions where where we find methylated cytosine. So in this particular region of chromosome 8, we have 6 individuals that have been exposed to ambient carbon dioxide concentrations, 6 individuals that have been exposed to elevated concentrations, and you can see that there's distinctly more marks here and fewer down here, indicating an epigenetic memory that has formed between the trees that are under elevated conditions relative to ambient conditions. Just to give a little bit more of what we've looked into in this region, I compared the regions, the DNA in this region, to other plants, animals, everything else that is available in a large database online. And perhaps unsurprisingly, we find lots of similarities with other Quercus species, so other oak species, because we have an oak tree. However, we do also find a fair number of similarities with the European beech tree. What does this mean? We're working on it. We also find that there are actually no genes in this specific region that is differentially methylated between ambient and elevated carbon dioxide levels, but we do find 207 transposable elements, which are jumping genes, which jump through generations within the oak trees. What does this mean? We're still working on it. So now I'm going to move on to talk a little bit about music and sonification. So sonification is a process where you are transforming a data set into sounds. For the purposes of artistic data sonification, this process can be split into three strata. At the lowest strata, at the lowest level, you have generative sonification, whereby you are determining the parameters, the pitch, the amplitude, the characteristics of the sound that is being produced, and faithfully converting the data using these sets of rules that you've generated into sound. At the second level, we have the elusive, which is what makes music music, the sublimeness of music as something interesting and good to listen to, which hopefully, for the purposes of this, is what engages the audience. Also importantly, as part of this, it includes ensuring the audience understands the data set which is being used to generate the piece of music, which hopefully I've explained to you in the last 10 minutes. And finally, we have the curatorial aspect, where the composer deliberately highlights the parts which are most interesting or salient or useful for the audience in order to convey the message, so that you don't have to listen to 800 million bass pairs worth of sounds. And just to go into a little bit more detail about the mapping function, or the process whereby the data set, which I presented to you earlier, is converted into the piece, I'm just going to talk you through a little bit of the code that was used. So just to explain, the process by which all this data, which is of course stored on a computer in the university supercomputer, is also computationally used. The rules, the generative and the elusive and the curatorial methods used were all done computationally, which is why I'm able to show you a little bit of the code which defines the logic of what actually has happened. So here we have chromosome 8, which, as I showed before, was maybe the part where the most interesting scientific developments are currently ongoing. We take some of the data headings from the differentially methylated regions table, and they were processed and assigned the different contexts. So the different contexts that were found in our data set were assigned slightly different chord sequences and methods of generating sound. What do I mean when I say different methods of generating sounds? Well, here is an example. So in this particular case, for example, it takes the input and outputs a sine wave with a particular lag modified by a random number at a particular frequency and with a particular amplitude with some panning, which adds some fading at either ends. And you can imagine there are lots of other slight variables along these parameters which can be tweaked or not. And essentially these blocks then allow customization of the mapping function, the way that the data set is then converted into sound. And to give curatorial information, it is sampling from the different contexts slightly differently, because biologically this is of interest, and it focuses on some of the chromosomes that we are more biologically interested in. And just to give some more information about the piece, which starts like this, it's a composition for a string quartet, the Ligeti Quartet, who were amazing in their performance of this piece. The Each of the four pieces in the quartet were sort of went through this kind of process with some slight modifications for musicality, and the whole piece was constructed into 6 parts with some echoes in order to invoke the idea of memories throughout the piece. So 2 points that I found to be very interesting about this piece. Firstly, the composer deliberately kept some of the outliers that normally scientists would have loved to remove from the data set. And this was actually musically very interesting, and throughout the piece you'll hear some high notes interspersed throughout, which represents the outliers that were part of our data set. And secondly, the feedback from the Ligeti Quartet was that they found this piece very challenging to play, in part because the traditional locations of where the beats are in classical music is completely thrown out in this piece because it is a sonification of the oak epigenome, and that made it challenging for them to keep their rhythm throughout. So, before I show everybody the piece, I'd like to thank Scott Wilson, who was the composer in this, Marco Catoni and Rosa, who both were very helpful in ideas and development of this piece, as well as the Ligeti Quartet, who were amazing in playing this piece. I'm just going to leave this QR code up, which is a link to the piece if anybody is interested. And then I will play in however much time I have left, a couple of minutes of the piece.